Ready, Anthony? Can we start? Wonderful. So, buenas tardes, good afternoon, everyone, and welcome back to our BP Endure virtual seminar series, Champions for Diversity in the Neuroscience. And today we have the pleasure of having Dr. Anthony Burgos-Robles, Assistant Professor of Neuroscience at the Department of Biology at University of Texas, San Antonio. Dr. Burgos-Robles did his PhD in biomedical sciences at the Ponce School of Medicine here in Puerto Rico under the mentorship of Dr. Gregory Quirk. And after that, he did a postdoc in neuroscience at Massachusetts Institute of Technology. He did two postdocs, he told me, one with Dr. Goodson and also with Dr. K-Tai. And Anthony's career path has focused in neuroscience research to examine the neuromechanisms underlying negative emotional states, such as fear, avoidance, and anxiety, using animal models. And specifically, he has identified several neurophysiological mechanisms occurring amongst cortical and subcortical brain circuits that are crucial for the dynamic modulation of behaviors associated to these negative emotional states. So, and currently, in his new laboratory at University of Texas, San Antonio, he is trying to address important issues in order to understand how these brain circuits get affected by repeated exposure to physiological stressors. So today we'll have the opportunity to listen about his most latest work and also show us how a young neuroscientist from Puerto Rico can also transform many lives of our students from the what we will call diaspora. So Anthony, thank you for being here with us, and we look forward to your talk.
All right, can you guys hear me clearly?
Yes.
All right, so let me share my screen here. I would like to first thank you for the invitation. It is always an honor to give talks to the Puerto Rican neuroscience community. I understand that also the crowd today is has variety of students and faculty members as well. So I'm assuming that there are no experts, but also there may be experts. I heard yesterday from Greg Quirk that he was joining, and also from Christian Bravo, which are experts in the topics that I'm going to be talking about today to you. So again, it is always an honor to share my data and my science with the Puerto Rican crowd, because I know that by sharing my experiences also in neuroscience and during my training and the success that I have had, I imagine I could inspire at least a few of those young minds in the island. So let me share my screen here quickly. Basically, what I would like to do today is to talk to you about some projects that I have done throughout my training in the Quirk Lab, as well as at MIT during my postdoc training, just to give you a sense of what kind of research you can do in these distinct environments that, although they are very distinct environments, like basically you can bring science and ideas that you have cultivated in the island all the way to the Massachusetts Institute of Technology to try to continue and get enhanced training for the further development of your projects and ideas. So,
Anthony, it
yes.
would be good to put it in slideshow.
Yes, I'm gonna just do that now. All right, and
Thank you.
after I talk about the a couple of the projects that I did during my training, I will also highlight a couple of new projects that we are working on in my lab related to neural circuits controlling fear, avoidance, and safety. So what do I mean by all this? So as you may imagine, like the emotion of fear, fear is just a natural, powerful emotional response in the presence of danger, right? You may think that having too much fear is too bad, but actually fear reactions are crucial, are very important for survival and safety. So the thing is that it depends on the situation, right? You may imagine that, let me use my laser pointer here. You may imagine that if in the presence of immediate danger, the first reaction that typically occurs is, something that looks like freaking out, which we term freezing, because, freezing uh, refers to the complete immobilization for a period of moment in response to something that is dangerous or a sensory cue that predicts the danger, right? So during the first part of my talk, and I will focus on condition freezing reactions and which brain regions play roles in such a function. Before I continue, you may imagine also that depending on the situation, in the presence of danger or the sensory cues that predict the danger, if the individual can assess, evaluate the environment and is able to detect options that may indicate safety, right? So, for example, if you can run away, then that will be initially a secondary response, which we term avoidance. So we don't call it running, we call it avoidance, right? Because the individual is trying to avoid or to escape the punishment that is upcoming. So again, for the first part, I'm going to focus on condition freezing responses, which was work that I did during my thesis in the query lab, initially in Ponce, and then the lab transition to the UPR School of Medicine, in which we were using these models, rat models, to investigate danger or threat detection and fear-related responses in the rat. So how does this go? So we term this either Pavlovian or classical fear conditioning. During a classical fear conditioning paradigm, there are stimuli that get associated to produce a response. In this case, the CS is, is indicating a conditioned stimulus. For example, a sound that doesn't mean anything initially, so the animal is showing no freezing reactions. But if that sound that initially is novel and it doesn't produce any freezing or fear-related responses, if it is paired with electrical shocks, for example, which is something aversive, something that the animals don't like, basically that as training progresses, those CSUS or conditioned stimulus, unconditioned stimulus pairings will start generating fear-like responses. In this case, conditioned freezing. The animal is gonna freeze, right? So during test, and in this case, it was 24 hours later, when the animal just listens to the sound that initially didn't predict anything, now the sound, the tone is predicting the upcoming electrical shock. And you may imagine that, if the animal learns very well, that the animal keeps showing these fear-like freezing responses compared to animals that receive that receive tones and shocks, but in an unpaired manner, so that there is no association in between. So the animal is not presenting condition freezing responses to the auditory cue. This is just an example. Let me see if I can remove this so I can play the video. This is just an example, one rat within the apparatus in which basically the apparatus is equipped with a speaker that we actually flagged in the video with infrared lights here to indicate the presentation of the auditory cue, and then we deliver mild electrical shocks through the grid floor, which produces condition freezing reactions. And this is just an example for you to see. So prior to the tone presentation, the animal is just exploring the cage, but you see how immediately the behavior of the animal change in the presence of the auditory cue that predicts the shock. The animal is going to receive the shock, yeah, right at the end of the cue presentation. But you see that, immediately after receiving the shock, then the animal just keeps doing its thing, exploring the cage. So this is why we term this a conditioned stimulus, because and a conditioned response, because the response is conditioned to the stimulus that predicts aversiveness. So using this paradigm for the last 3 decades, basically, many scientists around the world have shown several key brain regions involved in the learning of the sound shock association. For example, the amygdala, this is a brain region in the medial temporal lobe, which we know that puts together the association in between the cue and the threat, in this case, the electrical shock. But also many studies have shown other brain regions like the hippocampus, which we know that is important for processing contextual information. So you may imagine that not only the sound is a predictor of the shock, but the animal is in a specific location, in a specific cage environment. So the hippocampus should play a role in the processing of these contextual cues, so that the hippocampus can also integrate that kind of information with the other brain regions to generate fear reactions. We also know that regions in the medial prefrontal cortex, and this was, a lot of work done in the Quirk lab, since subregions. I think I lost you for a second. Sorry about that. My computer has some kind of hiccups that freezes for a little while. I was saying that in the Quirk lab, since the lab was in Ponce, uh, we were investigating distinct subregions of the medial prefrontal cortex. In this case, termed as the anterior cingulate cortex, prelimbic cortex, and the infralimbic cortex. And basically, a lot of studies have shown that these medial prefrontal regions are important for the regulation of the actual fear-like response, in this case, phrasing. So for example, we know that more dorsally located regions like the cingulate cortex and the prelimbic cortex enhance fear responses, whereas the infralimbic cortex inhibits fear responses, especially by inhibiting output from the amygdala. This is just to highlight, before I show you some of the data that I collected in the Quirk lab and it got published in the Journal of Neuroscience, investigating neural signals in the distinct brain subregions. So, a lot of people, including Greg Quirk, John Ledoux, Denis Paré, and many others around the world, basically stick electrodes within the basolateral amygdala, which is the brain subregion, or the amygdala subregion, that is important for making the cue shock associations. And before the animal received fear conditioning, neurons did not produce much signaling to the auditory cue that predicts the shock. But as the animal learned, after learning, when tested, neurons in the basolateral amygdala showed this large potentiated response to the cue that predicts the shock. But what was interesting always to me as a student in the Quirk lab was that the time course of the response of the neural signal within the amygdala, which was very brief in this case, for this specific amygdala neuron, that response only lasted for perhaps a couple hundred milliseconds. Notice that the tone length here in these experiments was only 2 seconds. So the question I asked while I was in the Quirk lab was to investigate brain regions within the medial prefrontal cortex, which we already had an idea that were playing a crucial role for fear expression. So what I did is that I stick electrodes, especially in the prelimbic subregion of the medial prefrontal cortex, and recorded neural activity while the animal was performing the fear conditioning paradigm. And these are just some of the findings we observed in this, we obtained in this study, in which in gray here is plotted the freezing response. So prior to the cue onset or the tone onset, the animal was showing no freezing response at all. But when the cue was presented, the freezing behavior increased drastically and lasted for the entire duration of the tone, in this case for 30 seconds, and even outlasted the key presentation for a little bit, decaying gradually until reaching baseline levels. But what you can see plotted here in the blue trace is the response of one single prelimbic subregion neuron. And as you can see, there is a very high correlation in between the behavioral response of the animal and the neural and the response of this neuron. Basically, these are just additional examples to show you that it wasn't a sporadic phenomenon that we observe in only one neuron, but actually around 25% of the neurons in the prelimbic subregion show this profile of response. Sustained response that correlates with the sustained behavioral fear-like response of the animal, in this case, freezing. So after I graduated from the Quirk lab, I was interested in going into the mainland to get some postdoctoral training. And as Carmen Maldonado mentioned, I first went to the Goosen's lab, in which I did some training on stress models in using rats, but then I wanted to learn more sophisticated techniques, such as optogenetics, which was initially developed in the Carl Dicerot lab at Stanford, in which basically specialized proteins that react to specific wavelengths of light, for example, either blue light or yellow or red shifted light. When these proteins are shined with this specific wavelength, they open a pore, and then they can allow flow of ions across the membrane so that they can change the activity of neurons if you're expressing these what we call opsins in specific brain regions or specific neural populations. The two of the mostly used opsins are term channel rhodopsin, which in response to blue light, it opens the pore, it allows sodium entry into the neuron, and that activates neural activity. Whereas arch-rhodopsin is important for inhibiting the neural activity because it removes protons from inside the membrane, from inside the neuron into the extracellular compartment. That leads to inhibition of activity in the neurons that you're expressing these opsins. So in the laboratory of Keita at MIT, what I did is that I wanted to answer the question, can fear responses be artificially modulated by selectively manipulating connections in between the amygdala and the prelimbic cortex? And this specific question was asked because, again, we knew that the amygdala is important for putting together the cue shock association, and the prelimbic cortex was important for outputting a signal, a sustained signal that can maintain fear responses for prolonged times as the cue is presented. So in this case, this AAV virus containing the genetic code for channel rhodopsin was infused in the amygdala, and after some time, to allow expression of the viral construct and the opsins, all the way to the axons of the amygdala reaching the prelimbic cortex, then animals were implanted with an optical fiber and that were coupled to specific of lasers of specific wavelengths. And then I was able to stimulate those axons to enhance the synaptic transmission in between the amygdala neurons and the prelimbic cortex neurons, right? By the same token, I did the same strategy, but instead of expressing channel rhodopsin, I expressed arch rhodopsin, and instead of delivering blue light, I delivered yellow light so that I could inhibit synaptic transmission from the amygdala to the prelimbic cortex. And here are some of the findings that I obtained from the study in the Thai lab. So first, what you can see here is modification of the classical fear conditioning paradigm, in which actually I wanted to integrate other things, other components in the paradigm to include an appetitive component or a reward-seeking component. In this case, what is represented in purple is trials in which the animal receives a specific cue we use either a light or a specific frequency of tone that was paired with sucrose that was delivering this plate. I'm gonna play the video here so that you can see what the behavior of the animal is. So the animal is exploring the cage. This light will indicate the cue that predicts sucrose, and you see that the animal immediately went into the port to retrieve the sucrose. And again, that was specific for the light cue, because when we measured the time that the animal spent in the port, the animal wasn't looking in the port during the pre-cue period, but when the light cue was turned on, then the animal immediately entered the port to collect the sucrose, because especially these animals were really well trained. But in orange, what you can see here is other tones that are intermingled with the reward tone or the reward cue, which we term the fear cue, which is a different tone, a different cue. In this case, it was a light versus a tone, and the animal and the tone was paired with a shock. And what you can see is that the animal is not doing much, just exploring the cage. But when the tone was turned on, the animal is showing this freezing. So having this clear distinction in between reward-seeking behavior and fear and conditioned fear response that is very specific for the key, the specific cue that is presented, then we were able to start stimulating or inhibiting the amygdala pathway, the amygdala to the prelimbic cortex pathway, to see if this neural pathway is important for mediating this kind of responses. So what you can see here is a representation, picture of prelimbic or medial prefrontal tissue, in which the prelimbic cortex is outlined with the dashed white lines here, and the optical fiber was supposed to be placed here, through which we shine blue light to stimulate the axons arriving from the basolateral amygdala. And what you can see here is that every time that the animal was presented with the cue that predicted the shock, and we stimulated the amygdala pathway into the prelimbic cortex, there was a significant enhancement of fear reactions, which wasn't observed when in animals that were just expressing yellow fluorescent protein control fluorophore. We observed, very importantly, no changes in reward-seeking behavior, meaning that this pathway is biased towards modulation of fear responses. By the same token, when we did the inhibition of this pathway, what we observed is a significant reduction in fear responses, in condition freezing responses, only during the cue that predicted the shock. Again, no changes to appetitive or reward-seeking behavior. Again, what these two findings say is that this amygdala to prelimbic pathway is both sufficient and necessary for the modulation or the production or generation of condition freezing responses. So this is just to summarize these initial findings that I obtained during my training at both Puerto Rico and MIT. And basically, we know that just by manipulating specific pathways within in between specific brain regions, we are able to modulate conditioned or learned behaviors. So what I would like to do is to use the remaining of my time to talk a little bit about the projects that we are conducting in my lab at UTSA, which are not mainly focused on the condition freezing responses, but in avoidance as well as safety seeking behaviors, because what we are trying to do is to develop new paradigms, or at least adaptations of paradigms that we know that work for examining active avoidance responses in the presence of danger. And let me start talking about the first paradigm that we are doing. Actually, before I talk about the very first one, I have to say again that our adaptation of a new paradigm is it has been inspired by more recent work in the Quirk Lab in Puerto Rico, in which animals, more than given the option to obtain food reward and the options for conditioned fear responses by pairing auditory cues, or in this case term, a tone warning signal that predicts shock, animals are also given options for escaping or avoiding the punishment, the shock punishment. So in this case, what you can see is that the animal is collecting food, it's actually pressing a lever for food, and then when the sound comes on, the animal runs to the platform so that they don't get the shock, the electrical shock. What you can see here is that in this case, this is representing that the animal successfully avoided getting the shock, right? Because the shock is not delivered immediately with the warning signal. The shock is delivered sometime after the warning signal has been playing, right? And what you can see in this graph, I'm sorry that is a little complex, but it contains information about the distinct behaviors that the animal is doing. So the animal is initially pressing a lever for food, right? Plotted in green here, but as the animal starts getting tone shock associations, then reward-seeking behavior drops dramatically, right? With a lot more training, then it seems that animals are resuming appetitive seeking. But what is important here is that the appetitive seeking behavior is being reduced because the animal is getting tone shock pairings, right? And here plotted in red, animals are showing condition freezing responses initially, right? That then it decays significantly. Why are condition freezing responses decaying significantly? Because the animal is also learning that there is an option to escape the shock. So as learning is progresses over many days, what you can see is that the time that the animal spends on the platform during the warning signal is significantly increasing as the animal is learning to avoid the negative punishment, right? So while we are using this as an inspiration, I have to say that in my lab, we are using mice. So we tried something similar initially to examine neural pathways and brain regions involved in this kind of learning. But the thing is that, first of all, mice are not as smart, I would say, at best, to learn all these things during these complex tasks and complex behaviors. So basically what we did is that is supposed to give animals an option to seek reward. We are manipulating the time in which the platform is present during the task. And this is work developed initially by Ryan Wood, a student, a rotation PhD student that rotated in my lab, and also is being followed by my graduate student, Maria Garza, in which mice are receiving tone shock pairings. And you see here the speaker to deliver the tones and the grid floor to deliver the shocks. But what happens is that, which is different from the Quirk Lab studies, is that the platform is not always present so that the animals can climb and just stay there forever. Instead of that, we make this platform a dynamic option, meaning that we can insert the platform or remove the platform at different time points. So this is how the task goes. So prior to the warning tone or the warning signal, we insert the platform, then the tone is presented and the food shock is presented. After the tone and food shock terminate, then we remove the platform. So what that means is that the animal learns to step on the platform only when necessary, right? Only when the warning signal that is predicting the shock is occurring. During the intertrial intervals, so that means in between warning signals, the animal needs to step down because anyways, we are removing the platform. So let me again switch here. And I would like to show you a short video. So this is an example of a well-trained animal. And as you saw here, so the platform is retracted, then the platform is inserted, and at some point after the platform is inserted, this infrared light is indicating the presence of the tone. And as you can see, the animal didn't go immediately to the platform because this is a well-trained animal. At some point after Q onset, the animal climbs on the platform and the shock is delivered right after. So that means that at least during this specific trial, this animal successfully avoided getting the shock punishment. And you saw that sometime after the tone shock terminate, then we remove the platform and the animal obviously needs to step down off the platform. So this means that it is highly likely that animals develop an active avoidance response that is signaled by the tone that predicts the shock. And that's exactly what happens. So this is some data from a pilot study. Basically this experiment contains around 20 animals. What you can see here, what we are plotting is percentage of time that the animal spends on the platform during the tone presentation, during distinct training sessions that occur in a daily basis. In black is the very first training session in which you can see that animals didn't show much learning. So most of the time during the tone that predicts the shock, the animals were not climbing on the platform that much. So they didn't realize that they can avoid the shock until the second session on red here, in which you can see that during the initial trials, that actually we didn't deliver the shock. The animals showed no learning whatsoever, but with further training, with further, again, platform shock, platform tone and shock pairings, then the animal shows us a significant and gradual increase in time spent on the platform, because finally, by the second session, animals realize that they can avoid punishment. But what you can see is that even with further training, we did up to 5 sessions, animals keep showing a significant increase in active avoidance responses by climbing on the platform to avoid the shock. And as time on the platform is significantly increasing with training, what you can notice here, this is a comparison of condition freezing responses during the first session, in which condition freezing responses were high, meaning that the animals learned that the tone predicted the shock. But as the animal is learning active avoidance responses, then those freezing responses significantly reduce. Just similar to the studies in the Quirk lab, even in this case without the option for reward-seeking behavior in between trials. So what we are also examining is the latency, or the time that it takes the animals to climb on the platform. And as you can see here, this graph is representing the time from tone onset that is taking animals to climb on the platform during the training sessions. And as you can see here, during the first session, especially when the animals were getting shocks, basically, in average, animals were not climbing onto the platform until the very last second, right before shock presentation. So actually, many animals were still getting shocks. But as learning progressed, what you can see is that animals learn to climb onto the platform sooner and sooner and sooner, meaning that they are learning that, again, the tone is the main predictor of the shock, not the platform, because they are not climbing sometime prior to tone onset. And that, again, because of reduction in freezing responses, they are enhancing their and improving their ability to avoid punishment. So what you can see here also is that we are asking more sophisticated questions. So for example, what happens if the animal learns to avoid the shock with only one platform in a specific location of the cage, but then we start tricking the animal by presenting the platform in the opposite corner. So this is happening here after 5 training sessions. So we teach the animals to climb in this platform first and then switch to the other platform. What happens is that initially, with the initially trained platform, animals are performing very well, but then when we switch to the new platform, animals show a significant decrease in avoidance response. When we switch back in the next 5 trials, animals are showing a very nice, robust, active avoidance response. And then with the new platform, again, alternating, animals are performing much better than with the very first bout in which we presented the new platform. So that means that this immediate learning profile is indicating that the animals are utilizing the memory that they acquire initially during the task to be able to adapt their responses to the new demands of the task, right? So looking through the literature, basically there is a phenomenon term schema-like learning, and that's from the Hippocampus field that says, again, that previous memories basically can help for speeding up learning of new demands in the environment. But again, like that has been a concept mainly investigated in the hippocampus and reward-seeking fields, not in the aversive conditioning field. So in this project, basically, we are looking at the role of the distinct brain subregions in the medial prefrontal cortex, especially the anterior cingulate cortex and the infralimbic cortex to determine what is their role in this kind of learning. By the way, I have to mention that just yesterday, maybe a couple of days ago, my graduate student showed me a new piece of data in which she was inhibiting the infralimbic cortex using optogenetic manipulations. And what she did was train animals while inhibiting the infralimbic cortex, and animals showed a significant reduction in or impairment in the acquisition of active avoidance responses. And then during the session 5, when animals were tested just with the platform and the warning signals, animals that got IL inhibition were not climbing onto the platform, meaning that the infralimbic cortex, at least, plays a significant role in learning active avoidance responses with dynamic platforms that are not always present. So that was one of the projects that we are working in my lab. The second project is a little different. So in this case, we are asking, what about other forms of threat, right? Because, you as, younger students, younger minds cannot perhaps conceptualize very well why are you teaching animals to be afraid of an electrical shock if that's not a very naturalistic phenomenon, right? So there is a new project that we are conducting in my lab in which we are using thermal threats. In this case, what I mean by this is that we can use either very cold or very warm temperatures in order to punish animals, or at least pair those punishments within their environment. And we are asking questions about like, are animals able to learn to avoid or at least do safety seeking like behavior based on what they are learning from previous days? So basically, again, so one of the main ones that we are using is very cold temperatures while animals are in a plexiglass apparatus. In this case, this is a combination of just normal wavelengths and infrared wavelengths pictures, in which you can see that there is a square-shaped plexiglass apparatus in which we add like visual cues on the walls, especially in one of the corners. You can see that it has a different patterns of symbols on the walls so that the animals can use those visual cues to locate themselves in the environment. And what we are doing here is that within this specific quadrant, we are reducing dramatically the temperature of the floor by just introducing either normal ice or just dry ice underneath the plexiglass and making this very cold. In this case, we are making that quadrant minus 5 degrees Celsius, whereas the rest of the apparatus, we warm it up using like warming blankets to keep it at a comfortable temperature. What you can see here is this is an example of a test animal that underwent this thermal threat avoidance paradigm in which animals during a training session for only 10 minutes, you can see that animals avoided this quadrant because it's so cold, perhaps because it's hurting their feet. And they are spending most of the time somewhere else, but not in that cold quadrant, which we are terming here the danger zone. What you can see here is, more data on this because we wanted to validate this. So we had a group of animals in which we introduced the cold quadrant versus a group of animals that the entire apparatus was at 30 degrees Celsius, right? And as you can see here is that most of the animals are spending time in the rest of the apparatus, but not in the cold quadrant versus those animals that didn't have the cold quadrant, they are spending time all over the place, right? So during the test session, what you can notice here is that we are testing the animals now without the cold quadrant to ask the question of whether they remember about this specifically previously danger quadrant. And as you can see here is that, again, those animals that had the cold quadrant, they were able to remember the specific quadrant that was warm and didn't spend much time in that quadrant. So I'm noticing that I'm running out of time, but what I would like to show you is just some pieces of evidence now that are leading for newer projects in my lab using this specific task. So for example, in this case, animals that didn't have the warm quadrant, they are showing spending some time in that cold quadrant. What we are also doing is that instead of using a cold quadrant, we are switching the temperatures here to ask the question of whether animals can learn about safety quadrants as opposed to dangerous quadrants. In this case, most of the apparatus is dropped to 5 degrees Celsius, while one of the quadrants we keep at a comfortable 30 degrees during the training session. Whereas during the long-term recall test, we just make the entire apparatus cold. But as you can see in this example, the animal is looking for the comfortable warm quadrant here most of the time during the test. So that means that they are learning that that quadrant specifically predicted the warmer, comfortable area. So this is just an example. One mouse for the very first minute during training, oops, sorry, they can learn this very quickly. So the animal is just exploring the apparatus during the first 10, 20 seconds, perhaps 30 seconds. And as you can see is that the animal sometimes is stopping within the warm zone, you see, and as learning progresses, the animal spends more and more time in that quadrant because it has a warm, comfortable feeling compared to the rest of the floor, which you can see from the video, that the animals cannot tell which is the specific warm quadrant based on the floor, but they need to use the cues on the walls to determine the warm zone. You see the video is still playing, but the animal is spending more and more time in that comfortable warm zone. So what we are asking now is questions about, like which brain regions play specific roles in the encoding of this safety memory. So we are wondering whether the hippocampus, which we know processes contextual information, whether if it plays a role in this. So basically, in some pilot studies, we injected animals with dreads, which is a specific receptor that combine this specific drug, the designer drug term clozapine and oxide, so that when the receptor is activated, it produces neural inhibition. And as what you can see here is a significant reduction in the time that the animals spend looking in the warm zone during training as well. Well, it's not so clear during the test session. We are still cleaning up these results based on histology. This is just, a clean example. Of course, we haven't go yet through all the animals that we ran so far. But we have indirect evidence also that the hippocampus is playing a very specific role, an important role in this, because when we use animals that have a genetic profile of Alzheimer's, in this case, the APP-PS1 animals, that when we get these animals through this safety learning task, animals have a harder time learning about the warm zone, but most importantly is that towards the end of the session, they finally learn, but the next day they don't learn where that warm zone was. Other questions we are asking are associated to social isolation stress. So, in light of all the COVID and pandemic in which, people are not only social distancing, but they are getting forced to isolate themselves, especially if they get the COVID virus, right? So what we have found so far is that animals can learn it very well, but they have a significant impairment during the recall test. We are doing further examination of this and asking questions about the specific involvement of distinct subregions of the medial prefrontal cortex, in this case, the prelimbic cortex. And I'm going to show you very quickly the data here because it's 450 already. So in this case, animals were initially group housed, then they were isolated for 2 weeks. So they were just a single house in their cages while they were in the animal facilities. And after those 2 weeks of social isolation, the animals were group housed again for a couple of weeks. And then we got them through the safety seeking task. At the same time, we are doing manipulations of the prelimbic cortex, especially we are inhibiting it using arshrodoxin, which is the opsin that induces neural inhibition. As you can see here in this graph, all groups of animals are learning just fine. In green, I'm showing the animals in which we are inhibiting the prelimbic cortex. And as you can see, the prelimbic cortex activity is not important for learning about the warm zone or the safety zone during the training session. But when that warm zone is removed during the test, animals that underwent social isolation stress, they show a full impairment in this memory recall. And I say full impairment because if you imagine, they are behaving just by chance. Nearly 25% of the time spent looking for the warm zone in this quadrant. So one quadrant out of 4 is 25%. So what is important here is that when we manipulate prelimbic activity, we can completely prevent this impairment induced by stress. So this is the stress group that during training, we were inhibiting the prelimbic cortex. And as you can see, these stress animals don't look anything like these stress animals. So these animals got their prelimbic cortex inhibited during training. So that means that we are able to both completely prevent long-term memory formation using this safety learning task, and we can also completely prevent this stress effect by manipulating one specific brain region. Lastly, by the same token, we are investigating the infralimbic cortex, and as what you can see here during the infralimbic inhibition experiment is that infralimbic inhibition didn't affect learning during the training session, but as you can see, during the test data, when the warm zone was removed and animals were allowed to explore and look for the previously warm zone, what you can see here again is the effect of stress in animals that were just expressing the control fluorophore, but animals that did not receive any stress, but we did infralimbic inhibition during training are showing the very same similar impairment as stress animals. So that means that just by inhibiting the infralimbic cortex during training, we are able to mimic the effect of social isolation stress in this safety-seeking paradigm. So, put together, basically what we are doing now in the lab is focusing on experiments in which we investigate the role of specific inputs within distinct prefrontal subregions, especially inputs from the basolateral amygdala, which we are just producing the animals now, as well as the input from the ventral hippocampus, which both send very robust projections to the medial prefrontal cortex, and we believe the medial prefrontal cortex neurons are utilizing that information from the amygdala, signaling danger or safety, and also information from the hippocampus, which signals, specific features of the environment in order to put together these memories for safety and avoidance of thermal threats. With that, my apologies for running just a little bit over time. I'm happy to stay for a little longer for questions, but before we proceed to questions, I would like to highlight and thank all the people in my lab, especially Ada Felix, which is a research scientist and lab manager in my lab, which is running all these experiments in thermal threats. Maria Garza, which is conducting the dynamic platform experiments, and some of the undergrads that are participating in the research projects in my lab. With that, I will be happy to answer your questions. Thank you.
Thank you, Anthony. That was very interesting. And I really very much, I'm very curious about how, it's going to develop this new paradigm for you from the standpoint of safety seeking. I think that is a very important phenotype within the emotional process that involves avoidance learning and also, of course, fear. I don't know, I haven't gotten any questions in the chat. Any questions that anybody wants to type in, if there's chances to type something?
I'm sorry, if there were questions in the chat, it's hard to, do the presentation and keep track of the chat.
Yes. Let me see if something shows up here. I haven't seen it in there. Well, okay, so because unfortunately with Zoom, they cannot turn on, the participants cannot turn on their mics. So that's the only problem. I don't know if Marimar, do we have questions in the Q&A or?
No, I don't see any, but if someone wants to turn their mic on, they could raise their hand and I could let them open their mic.
Okay, so anyone that wants to make a live question before we go, we still have 5 minutes.
Yeah, feel free to ask the questions now. Yeah, related to what you were mentioning about safety learning, we got inspired by some of the ideas in the field, but also because, the more I talk with my students, over lab meetings, we realize that research in safety learning is at best somewhat poor, I would say. I don't want to critique, some of the good research out there, but in this case, most of the research on safety seeking is using specific cues, like for example, from the standpoint of fear conditioning, right? So cue predicts something aversive, shock, right? Versus another cue that doesn't predict anything, that is considered by some studies as a safety signal, right? So because one is indicating danger, the other one is indicating no danger, right? But one of the things I've been discussing with my lab here is, that doesn't necessarily mean that that's a safety cue, right? Because it just may mean nothing, right? It means nothing, just as before pairing the cues and shocks, right? Before pairing the shocks with the cues, the cue doesn't mean anything. It's just an audible sound, that the animal just, stops paying attention very quickly, right? So we are coming up with newer ideas to try to uh, tackle this question about, how animals can learn about safety, especially, using more naturalistic threats like thermal threats. And
Yeah, I really like the design on the thermal tracks. That those were really cute and interesting for me. To coming from years of behavioral pharmacology and animal models, I thought that was quite interesting and very that had a lot of potential for many things. And so,
yeah,
uh-huh.
and you can perhaps remember about all this research on hippocampal involvement
Yes,
on spatial navigation, for
of
example,
course.
using the Morris water maze, right?
Yeah.
That's the very typical way to go if you want to examine like some kind of danger. In this case, like the animals, if they don't swim and find the platform, they sink, right? But in
Exactly.
this case, we got some kind of inspiration from the
Yes,
Morris water maze, but in this case, it's a completely dry paradigm, right? Animals don't need to swim, yet they need to be able to locate safe versus danger locations within the apparatus that they cannot really tell through visual indication of the floor, but they need to use the cues on the room, in this case, the apparatus on the walls, to locate themselves and say, okay, so it's this quadrant that it has the comfortable temperature versus the rest, it's cold and I don't like it. So we are basically very excited about these new paradigms, and basically we can now be able to answer more specific questions about all these neural projections, which we know that all these brain regions interact with each other during multiple other paradigms and fear learning. So let's see where the future takes us with us.
oh
With this.
yeah, I know it's going to be a very bright future, and we certainly look forward to your next publication on using these paradigms. And unfortunately, we ran out of time because it's 6 already, but we really appreciate Anthony for taking the time, for being with us this afternoon. Uh, I want to stress to the students that Anthony is an example of the possibilities that we all have if we really love science and neuroscience and want to establish independently our lab. Now that he's at University of Texas, San Antonio, we really want to send him the best of success because it's going to be a very successful career. We know that, knowing Anthony's performance till now. And thank you, and we look forward to future visits here in Puerto Rico. I hope when you come back to your hometown, you come and
Yeah,
visit us.
I haven't been in Puerto Rico since the pandemic started.
Well,
And
imagine. So you may have to wait a little longer, ¿verdad? Pero we look forward to seeing you again
yeah,
and talk about your your great science.
yeah,
And sure. Thank you, Anthony. Muchísimas gracias.
before we finish, I would like to mention that, like, and I'm highlighting here my email address. So anyone that is, especially from the students that is interested in the neuroscience program at UTSA, like, feel free to email me to gather more information. I can send you some more information, give you some more specific contacts from the, oh yes, it's in the chat right now. Thank you for whoever shared it. But we have an awesome neuroscience program. Not only me here doing behavioral studies like this, but there's also people that are more focused on the hippocampus and spatial navigation, learning about features in the environment, about manipulating specific populations of neurons, people that do like reward seeking and addiction learning. So there's a whole lot going on here at UTSA, and I hope, you know, like we can create, you know, a stronger bond basically to try to create these connections with Puerto Rico and UTSA and Texas to try to increase the number of Boricuas here in San Antonio.
Wonderful, wonderful. Great. That's a great goal. I appreciate that. And so I just want to end encouraging everyone to come back next week on Wednesday, because we have Dr. Jasmine Hurd, Director of the Addiction Institute at Mount Sinai and Professor of Pharmacological Sciences and Neuroscience at the Icahn School of Medicine. So that's going to be also a great talk. So we look forward to seeing everyone next week, and buenas tardes.